Okay, this is a video lesson over Unit 12, which is going to be discussing all the different types of mutations that can occur, both chromosomal and gene mutations. Make sure you're recording these notes in your journal and pausing and rewinding any parts that you need to do so. The first question we need to discuss is, what is a mutation? Well, a mutation is not typically what we think of being, because sometimes, like in film and movies and things like that, a mutation is something that causes a big major change in the person or the organism. Like, you think of the Hulk or the Mutant Ninja Turtles. When in reality, a mutation simply is any change in the DNA sequence. So when we are changing the DNA, whether we're changing the chromosome or we're changing the DNA itself, the genes, we are causing a mutation. A mutation has occurred. Okay, so let's start off by discussing chromosomal mutations. Okay, so remember that a chromosome is a way that the DNA is organized within the cell. Okay, so a chromosome is simply a condensed form of our DNA. So chromosomes, if they're duplicated, they look like an X. Okay, if they're unduplicated, though, they're going to look more just like a straight line. Okay, so we'll just draw it kind of like this. This will be our single chromosome. Okay, so in every human, all humans have 46 chromosomes total in their cells, okay, with the exception of gametes, which are the sperm and egg cells. All of your body cells have 46 chromosomes, okay, and each of those chromosomes codes for and has certain gene sequences or DNA sequences on it that code for specific proteins. Okay, so a chromosomal mutation is going to be a change in the chromosome itself that's going to happen and cause the resulting proteins to be made from that chromosome to be different than they would have been originally. So let's talk about 4 different types. The first type we're going to talk about is deletion. Okay, so deletion is just like what it sounds like. So let's draw our chromosome number, our chromosome. Here's our chromosome. To help us understand this, we're going to write some letters on our chromosome that represent genes that can be located on the chromosome. So we have A, B, C, D, E, F. So these are genes on our chromosome. Okay. Now, if a deletion occurs, what's going to happen is, let's say some sort of deletion occurs and this B is removed. Okay? This part of the chromosome is accidentally left out or removed from the chromosome. So that leaves our gene sequence on our chromosome to be A, C, D, E, F. Okay, so you can see here that it's clearly able to see that a deletion has occurred because that B is no longer there. Instead, we skip from A directly to C. So this would be our first type. This is a deletion. Okay, the next type of chromosomal mutation is going to be insertion, okay, or duplication. Those can mean the same thing. So let's draw our chromosome again. Here's our original chromosome. And we're going to have A, B, C, D, E, F. So if we have an insertion or duplication mutation on our chromosome, what's going to happen is, let's say we add in an extra C. So we'll say A, B, C, C, D, E, F. So you can see here that this C has been added in, this segment of the chromosome has been added into the chromosome. So we have added or duplicated a part of the chromosome. Okay, so this is our second type of chromosomal mutation. Our third type of chromosomal mutation is called inversion. Okay, so inversion means you're reversing the order. So here's our chromosome. A, A, B, C, D, E, F. So if we have an inversion occur, we're simply going to be flipping this order around. So this chromosome might read instead D, E, F, A, B, C. Okay, so we have simply the bottom has flipped with the top. Okay, so it's just like a little flip-flop and the order gets rearranged. So that is an inversion where we are reversing the order. The fourth type of chromosomal mutation is called translocation. Okay, so trans means we're moving somewhere. Location means we're moving to a new location. So here is, then this is going to involve more than one chromosome. So we have our first chromosome here, which has our genes A, B, C, D, E, F. And we're going to have a second chromosome that's going to have genes G, H, I, J, K. Okay? So in translocation mutation, what's going to happen is part of one chromosome is going to be removed and added to another. So let's pretend like this part here, JK, for some reason decides to come off of that chromosome and attach over here. So this is going to result in our first chromosome. First of all, it's going to get bigger in size because we're adding more chromosome to it. It's going to end up looking more, it's going to have 2 extra genes to it. And our other chromosome, chromosome 2, is now just going to be G, H, and I. Okay, so we have moved the part F, J, and K from our other chromosome has been moved over here to for our first chromosome and added to it. Okay? I'm sorry, just J and K are added. We aren't actually adding F. We're only adding J and K from our other chromosomes. Let's not get that confused. So we're only adding J and K. Okay, so those are our 4 types of chromosomal mutations. Now, chromosomes are the bigger picture. So that's how the DNA is organized on a bigger scale. But if we zoom in even closer, directly into our DNA sequence, we're going to have some different types of gene mutations. So these are mutations that are changing the actual sequence of our genes. Okay? All right, so the first type that we're going to talk about is called a substitution mutation. Okay, or a point mutation. These are the same thing. Okay, so a substitution mutation or a point mutation means that a base or bases are exchanged with another. Okay, so that is a substitution mutation. So let's work an example so you can kind of get an idea of what I'm talking about. So let's start off with a simple sentence. The dog ate the pie. Okay, notice that this sentence, all the words are 3 letters long. Okay, and I chose this sentence on purpose because when we think back to protein synthesis, how many letters at a time is the ribosome reading our mRNA sequence? You should say 3. So our ribosome reads 3 letters at a time. So this is going to be important because if we have mutations, it can change and affect the way the ribosome is actually reading our mRNA message. So we'll start with the sentence, the dog ate the pie as our original sentence. But what happens if instead of this D here, this D gets substituted for an H? So now our sentence reads, the hog ate the pie. Okay, so this D is the only change that we've made to the sentence is simply exchanging or swapping out that D for an H. But now our entire sentence has changed. Okay, so the dog ate the pie, we now have a hog that ate the pie. You can still read it, it still makes sense to you, and you still understand what it means, it just means something totally different. Okay, so the same thing is going to happen when we talk about what's happening in our DNA sequence. So let's write this original DNA sequence down. Right, so for our original DNA sequence, we're going to say our DNA sequence is TAC, CAG, GAT. Okay? I want you to pause the video and figure out the mRNA sequence and the amino acid sequence using your codon chart. Okay, so if you got your mRNA sequence, remember that in mRNA, T pairs with A pairs with U, C with G, C with G, A with U, G with C, G with C, A with U, and T with A. Right, so our amino acid sequence that you should have gotten, if you looked them up on your chart, was methionine, valine, and leucine. Okay, so this will be our original DNA sequence, and which codes for this specific protein, okay, these amino acids. So let's show what happens if we do a substitution or point mutation. So here's our mutation. Our mutation is going to be, we have our DNA, but now in our DNA it's going to read TAC, which is still the same, but instead of a C next, we're going to swap out that C for a G. So it's going to read GAG. Here is our mutation. GAT. Right? Let's now figure out, pause the video, take a second to figure out the mRNA and the amino acid sequence now. Okay, if you, once you do this, you should remember that mRNA pairs with, we should get AUG, CUC, and CUA. Right? Your amino acid sequence should read methionine, leucine, leucine. So now let's look back and compare this amino acid sequence, our mutated amino acid sequence, to our original amino acid sequence on the previous page. How do they compare? Well, originally we had methionine, valine, leucine. Now we have methionine, leucine, leucine. So you can see that that second amino acid is now different. Okay, so we now have changed the protein by simply changing out the one base, this G, by exchanging this G for this C into a G. We have now changed the amino acid that goes in the protein. So our protein is changed. Okay, so this is a substitution mutation. Now, notice that the amino acid after is still the same. So in a substitution or a point mutation, we're actually only going to be changing that one amino acid, okay, that's coded for by the changed codon. So the rest of the protein is going to remain pretty much unaffected. Okay, so the next type of gene mutation is called a deletion mutation. And a deletion mutation is a type of frame shift mutation, which will make sense in just a second. Okay, so in a deletion mutation, this is going to be when a base or bases are deleted from the DNA sequence. Okay, so we're simply deleting it. This is super easy to remember. Let's work out an example. So we're going to start off with our same sentence that we had before. The dog ate the pie. So in a deletion, we have to delete a letter. So we're going to delete the D. Okay? Now, when we remember our ribosome reads letters in only codes only in 3 letters at a time. So we have to write this in 3 letters at a time. So it's going to say V, O, G, and now we have an A next, so it's going to be A, T, E, T, H, E, P, I, E, followed by whatever letter would be after that in the DNA sequence. So we have shifted. This is a frame shift. Our reading frame has completely shifted because we have removed this one letter, the D, okay? So now our sentence reads the Oga Tet Hep IE, so whatever that means, okay? Because we had to move all the letters over since our ribosome reads only words that are 3 letters long, okay? Let's go back and look at our, let's rewrite our original sequence. So the original DNA sequence that we had before was TAC, CAG, GAT, and our mRNA, remember, was AUG, GUC, CUA, which gave us the amino acid sequence, methionine, valine, and leucine. Okay, so let's look and see what happens when we have a deletion mutation in a DNA sequence. So for our mutation, we are going to have a DNA that now reads TAC, and we're going to delete that next C. Okay, so now we're going to have A, G, G, A, T, and we'll just pretend the next letter is T. So in our mutated DNA sequence, what we've done here is we have simply deleted this C here, has been removed from our sequence, causing our reading frame to shift over. Okay, you can see how it's shifted over if you compare the 2 sequences. Okay, now let's pause the video and I'm going to give you a chance to figure out the mRNA and the amino acid sequence. Right, for your mRNA you should have AUG, UCC, UAA. Right? So our amino acid is going to be methionine as our start. You should get serine and then a premature stop. Okay, so if we can compare this to our original amino acid sequence, methionine, valine, leucine, we now have methionine, serine, stop. So that means that every single amino acid after that mutation has changed. Okay, so every amino acid after the mutation is changed. Okay, that's what happens in a frame shift mutation. Okay, every amino acid after that mutation is going to be changed, which causes a greater change in the resulting protein. Okay, especially in this instance when we have a premature stop. So our ribosome is going to think that it's done making the protein, even though it actually shouldn't be done. All right, our last type of mutation is called an insertion mutation, which is going to be the exact opposite of a deletion mutation. So an insertion mutation is also a type of frame shift, meaning we're shifting our reading frame that our ribosome is reading through. And the definition for this is a base or bases are added to our DNA sequence. Okay, so let's work our last example here. Now our sentence, we still have the dog ate the pie. But now what we're going to do is I'm going to insert an extra letter here. We're going to insert an A. So our sentence is going to read now, after this insertion, the A-D-O G-A-T-E-T-H E-P-I, and then E something something. Okay, so we have shifted now our reading frame in the opposite direction as we did in the deletion, all because we've added in this extra A here. Okay, So let's look and see what happens when we actually do this to a mutation. So if this occurs in a mutated DNA, our DNA sequence might read, instead of TAC, CAGGAT, we're going to add an extra A before that C. So it's going to say TAC, ACA. This is our extra base that's been added. Okay. Then we have GGA, T something something. So remember, if we look back over here, you can see that our original DNA strand was CAGGAT. Okay, so now we have simply added an A before that C. Okay, the resulting mRNA, I want you to take a moment and look up the mRNA and the amino acid sequence. When you look this up, when you figure this out, you should get AUG, UGA, or UGU, I'm sorry, CCU, A something something. Okay, our amino acid sequence should be methionine, cysteine, and proline, followed by something we don't know. Okay, so again, in the insertion, this frame shift mutation, we have shifted the reading frame, causing all of these amino acids after our mutation are now changed. They are different. Okay, so a frame shift causes a bigger effect on the protein produced. Right? Now, you might be wondering, why are mutations important? Okay, mutations are going to be significant from an evolutionary standpoint, because if a mutation happens to be advantageous to an organism, right, then that mutation is going to stick around and it's going to be passed on from one generation to the other, assuming that the mutation is in the hereditary DNA, which would be found in the gametes. We'll talk about more about that when we talk about meiosis. All right, so make sure that you have everything written down. Go back and rewind, pause, and rewatch any parts that you need to rewatch. And make sure I check off your notes to know that, so that I know that you have completed your video lesson.